All right, hello everyone. We're going to start into chapter 11. This is going to look at Mendelian patterns of inheritance and how genetics are passed on from parent to offspring. And we're going to be talking about this gentleman here in a little bit. His name is Gregor Mendel, and he's one of my favorite scientists. So let's get into it. Now, I've kind of moved the sections around a little bit. Hopefully 11.1, we're just going to be talking about genetics and the vocab of genetics. So check your PowerPoint image, just make sure we're on the same slide because I moved things around here. So gene, this is a section of DNA. It codes for a characteristic of your life. You can see here our genes earn a code for some characteristic. Now, there are different forms of these genes. You can see here we have like different eye colors or different hair colors. Basically, I give the analogy of like genes are like a shirt and the alleles are like the colors of shirts. So like everyone has a shirt on, but everyone might have a different form of that shirt or different color of that shirt. So basically, again, genes, section of DNA, they code for something in life. Alleles are just different versions of those genes. A genotype refers to the pair of alleles or the combination of alleles that we're going to find on these homologous chromosomes. So every allele is one letter. The genotype is the combination or the two letters here. So again, two letters instead of one. Capital letter will always be put first here, but that's about it. Phenotype, these are the physical characteristics of an organism. So you can see this is what is produced from the genes. You can see the phenotype, physical characteristic. This is caused by the genotype or the genes of an organism. Now we have two different versions of a genotype. We have homozygous and we have heterozygous. Genotypes that are the same are homozygous, big B, and little b. These are examples of homozygous genotypes. Heterozygous genotypes are genotypes with different alleles. So you can see big B, little b, these are heterozygous. I always love to talk about these pair of heterozygotes. That's just a little funny. I know they're bad jokes, but we have to throw them in here. Dominant alleles. These are alleles with fully functioning DNA that produce a fully functioning protein. Now, in your biology classes, you may have learned this as the stronger of the two alleles, and that is true, but this is the more scientific and more accurate understanding of what a dominant allele is. Recessive alleles are where DNA doesn't produce a fully functioning protein. It either has very little or no function at all, but it is represented by a lowercase letter. So dominant allele, fully functioning, stronger allele, capital letter, recessive allele. Weaker, it is producing a non-functioning protein, lowercase letter. Oh, we're going to stop right there then. Yeah, this is where we're stopping. All right, one thing I forgot to go over in this section is the law of dominance. And this is where when in a genotype, you have one allele that is dominant. So let's say it's heterozygous or homozygous dominant. That phenotype will exhibit the dominant phenotype. If an organism receives two of these recessive alleles, that's the only way they're going to exhibit the recessive phenotype. So again, as long as there's one dominant allele, it's going to be the dominant phenotype. When there's two recessive alleles, it's going to be the recessive phenotype. All right, moving on to 11.2, we're going to look at Gregor Mendel a little bit more in depth. But basically, Gregor Mendel Mendel changed how we looked at genetics and inheritance. Now, before Mendel, the blending hypothesis was accepted. And this is where there is a combination of the, or like a mixing of the parents' phenotypes, and that's going to be exhibited in the offspring. You can see here there's a mix or there's a blend of these phenotypes in the offspring. Now, this makes sense in some data. So you can see sometimes we do see a red and white flower, but it doesn't make sense for all of it, because if all flowers followed this blending hypothesis, you can see here, every flower would be only one color and we'd only have one. So it just doesn't make sense that the blending hypothesis is correct. So Gregor Mendel was an Austrian monk in the 1860s, and he was very educated. He went to the University of Vienna. He studied both, I think, botany and math. He was also a substitute science teacher, but he was really the first scientist to apply math to biology. I always think he looks like Dwight from The Office, but that's just me. So basically what he did was he looked at pea plants because they had really distinctive features, and they also reproduced pretty quickly. And now he worked for over 8 years on 28,000 pea plants, and he looked at certain phenotypes. And you can see what he did was he watched the he watched which plants would, he controlled which plants mated with other plants by using a paintbrush and taking the female and male part, the pollen, and collecting it and putting it to the female part here. So he controlled basically which plants had sex. And this allowed him to look at these crosses or these matings a little bit more specifically and look at their offspring and what they produced. This is what the garden of his looked like before, and this is what the garden looks like today, as you can see there. Now, the reason why he chose, again, these pea plants is because they had distinctive features. He could easily tell each of these different features. Now, he kept crossing true breeding plants, so tall with tall and short with short, until he got only tall and only short. Now, if the blending hypothesis was true, the offspring would all be medium, and then there would be more medium from that. Now, this is called a monohybrid cross. We're looking at a single trait, so like tall versus short. Now, what happened was, as you can probably tell, they weren't all tall. So the tall and the short, when crossed, they produced all tall plants. And that didn't make sense because they should be all medium. So what Mendel did then was he took these tall plants and crossed them, which, yes, it's a little bit of inbreeding, but in this case, they're plants, so it really doesn't matter. But what happened was he found a 3-to-1 ratio of tall to short. This was the F2 generation. And this perplexed him because he found the short plants again. He didn't know why. Now, he wrote about this in his data. You can see his data matched this 3-1 ratio. And it's not perfectly 3-to-1, but it's very close. And that's because, again, he took a lot of data. It took many years for this to look at this. And now, he wasn't sure if it was heterozygous or homozygous dominant, but he didn't know these words. Again, they didn't really know about cells yet. There was still a lot of genetics that just wasn't worked out. So if we looked at these crosses, what we understand is they were homozygous dominant and recessive. Now, I don't have this filled in, but basically what we see in the offspring is there are all heterozygous genotypes in these boxes. So each one of these is a heterozygous, which means they're all going to be tall. In the F2 generation, you can see here, we get the 3-to-1 ratio because we get a homozygous dominant, 2 heterozygous, and a homozygous recessive here. So in this case, we get that homozygous recessive back only in one of these 4 squares. So this is why it's a 3-to-1 ratio. Now, this is because these traits separate independently of each other. These alleles will separate during meiosis, and they're inherited independently of each other. And this is known as the law of segregation, where each gamete only can contain one of these factors, and they separate during meiosis. So basically what we're showing you here is these Punnett squares are like the sperm and the eggs, and the Punnett square itself is the chance of having an offspring. You can see these alleles are like the father's alleles, and these alleles are like the mother's alleles. You can see the boxes here are the possible offspring. Now, Mendel published his work, and he called it the particulate theory. Wasn't perfect, but it gets us on our way to understanding genetics. He isn't really known at the time as being this, monumental scientist. We look back and understand he was well beyond his years. But his last words were, "My time will come," because he understood how important his information and his data was. So this is where we're going to end with the 11.2. What did Gregor Mendel say when he founded genetics? Whoopee. Okay, bad joke. All right, 11.3 is going to look at dihybrid squares. Now, Mendel thought to himself, if these data support that the traits are inherited independently of each other and these alleles separate, now again, he didn't know any of those words, but he basically understood the thought behind them. But he thought, does this hold true for when there are several traits? And this is what he was observing then in the pea plants. He looked at 2 traits, and this is going to be a dihybrid cross. So before we get into a dihybrid cross, let's talk about the locus or the location of the gene on a homologous pair of chromosomes. So this is the locus. It's the specific location where these alleles are found. Now, you can have different versions of these genes. These are known as alleles, but they have to come on homologous pairs of chromosomes. Now, with the law of independent assortment, remember that each of these factors are going to separate independently of each other. And that basically means that we're going to create a combination or a set of combinations of these chromosomes. You can see in this in these cells, any one of these combinations is correct. You can have any one of these maternal or paternal sides independently assort during metaphase, or I'm sorry, meiosis one. It's during metaphase one. So any one of these combinations is correct. What does that mean for us? So as you can see here, we have a number of possibilities where, let me go back here, where it could be big A, big B, or big A, little b. It can happen either way where each of these is independently assorting from each other. Now, again, what does that mean for us? Before we get there, this can also occur in chromosomes where the alleles are the same. This is just when crossing over occurs. Okay, so this is crossing over or independent assortment can be the reason why we have to show both of these. So if we look here, we have to independently assort each one of these, or we have to separate each one of these alleles because this is a genotype or two genotypes, and we have to only allow one of each of these alleles to be passed on from parent to offspring. So our first gamete is big A, big B. Our second gamete is big A, little b. Our third gamete is little a, big B. And our fourth gamete is little a, little b. So these four gametes, or our four combinations here, are possible just from this one parent. So what I'm going to have you guys do is a dihybrid cross, which is next on the on this slide. You're going to put these gametes here. So it's going to be big A, big B, big A, little b, little a, big B, and little a, little b here. And then the same, let me go back. The same on this side. Now, when you combine them in these spaces, when you combine them in these locations, you got to match the A's. So, sorry, it's going to be big A, big B, big B. I'm sorry, big A, big B here. And then you're going to combine the two genotypes here. Now, I'm going to do this in class, but I just wanted to briefly kind of let you set this up and try to do a dihybrid cross. All right, the second part of 11.2, we're actually going to be using math here. And don't be scared because this math, it really isn't that bad. Looking at a probability problem, what's the chance of rolling snake eyes with a pair of dice? What's the chance of rolling a 1 on a dice? That's 1 out of 6. So what we have to do is we have to multiply them together. So 1 out of 6 or multiplied by 1 out of 6 is a 1 in 36 chance of rolling snake eyes. So that's the chance of rolling a snake eyes with a pair of dice. So understanding this probability, we have to kind of look at it as maybe not rolling dice, but flipping a coin. So what's the chance of receiving a big R or a little r, or a heads or a tails in this case? Now, I'm going to show, this looks really complex, but it's really not. It's going to make our dihybrid crosses really easy. So what's the probability of two heterozygous genotypes producing a homozygous recessive offspring? You might be able to do this in your head, but mathematically, let's take a look. There's a 1.5 and 1.5 chance. So our probability is 1/4 of producing that little b in this case. This is an easy kind of just starter off, thinking about probability. We have this location here. But if we can make this into a dihybrid, it makes it a little bit more difficult. In a cross between, you can see the heterozygous. What's the probability of having the little a, little b, little b? Well, if we do the two monohybrid crosses, there's a 1 in 4 probability of having a little a, and the the little b, there's also a 1 in 4 probability. So it's a 1 in 16 because we just multiply them together. And if we did that dihybrid Punnett square, which we we did it before, we found it was a 1 in 16. So this is a way that we don't have to make those dihybrid crosses. We make them a lot easier. So we can do this with a trihybrid. So taking a look here, what's the probability of getting this genotype with these? Well, if we look at it, there's a 1/2 chance, or 1 out of 2 getting this. There's a big B, there's another half, and a C, there's a 1/2 if we do the monohybrid Punnett squares. It means it's a 1/8 probability of producing this genotype. So all you have to really do here is just find the monohybrids and multiply them together, and you get this as your ratio. So this is how we make dihybrid Punnett squares a lot easier, or even trihybrid or anything other above that. There are some more examples here. I'm not going to go through them, but this is the end of 11.2. 11.4 is going to be a kind of short section, but basically we're going to go over things called autosomal recessive and autosomal dominant like diseases or disorders. So in an autosomal recessive disorder, individuals have to receive both recessive genes to be affected. You can see in our these aren't really Punnett squares, but in our inheritance patterns here, you can see that the only person here that is affected is this person here that has the 2 recessive alleles. These individuals can pop up in a population. Now, that's usually rare, but these will pop up when the parents are heterozygous. So that means that you have one of each of these alleles. You can see that 3/4 of the time, the individuals will not have the disease. However, when they are heterozygous, we like to call them carriers. So they carry this disorder, but they don't actually have it. The only people that have this are little r, or the homozygous recessive in this example here. Now, this tends to skip generations because, again, the heterozygous, you don't have it in this generation, but in this generation, you do it 25% of the time. Now, the thing to remember here is autosomal. These are chromosomes 1 through 22, and that means they're not sex determining. So this is going to be equal in males and females. Yeah, so let's take a look at an example, CFTR. So this is the gene that causes cystic fibrosis. It's found on chromosome 7, but basically the gene doesn't produce a fully functioning protein. So there is something that goes wrong. I'm not going to go too much in depth here, but the quote unquote normal gene is the dominant. The recessive gene is people that have the cystic fibrosis, and it just produces a protein that doesn't work right. So people that are heterozygous don't have this, but people that are homozygous recessive do. Now, oh, by the way, CFTR, or that the cystic fibrosis can be deadly if not treated. So these are like kind of deadly diseases, and if they go untreated. The other type is autosomal dominant. This is when you need only one of the alleles. So you can see here big A, or big A, little a. As soon as you get one, you get this. This is, again, very rare because if you do have it and you're affected and it's a deadly disorder, I mean, it's going to evolve out of a population. So again, these are genes found on chromosomes 1 through 22, not the sex chromosomes. So you can see an unaffected mother, affected father. They're going to pass these on more often because once you get it, you have it. Huntington's disease, this is an example of a autosomal dominant. The thought there is, if it's deadly, why would a person still inherit it if it's evolving out of our population? These diseases usually hit the person, or they develop later on in life after the person has already given birth. So that's how they can still persist in a population. But this gene affects the brain and how it functions. I'm not going to go too much in depth with these, but this is all we're going to go over for 11.4. Hello, everyone. This is going to be 11.5 genetic recombination. But basically, in this chapter, we're going to look at how crossing over really affects the genetic recombination in what we see in chromosomes. So, talking about these two gentlemen again, we see them working with fruit flies in the early 1900s, and we understand that chromosomes are going to carry our inheritance or our genetics, or our DNA, and they're going to be the chromosomes are going to be passed from parent to offspring. Again, this is about 50 years later after Mendel, and these guys were like, hey, Mendel was actually kind of right about what he was learning about with his pea plants. So remember, what they found in their fruit fly experiments was these two alleles being inherited by or they're located on the chromosomes. Now, again, they don't know what's in these chromosomes. They don't know what alleles really are, but they know something is being passed from parent to offspring in these chromosomes. Along comes this guy named Thomas Hunt Morgan, and he studied fruit flies even more. He analyzed the characteristics that we see in these fruit flies, kind of like that how Mendel looked at the characteristics in these pea plants. Now, looking at these pea plants in the dihybrid Punnett squares, he started noticing that the inheritance patterns didn't always make sense. And I'll show you what he thought he should find and what he did find. So through independent assortment, we understand this. This was in your packet. Yeah, we should have a chance of having all four of these. So big A, big B, little a, little a, big B, big A, little b, should all be possible. So we should get that 25, 25, 25, and 25%. So that's why we do the dihybrid Punnett squares. Yes, this should happen, but it's not what he found. So he actually found them. It wasn't even close. We were doing these labs this past week where we were looking at how close our data was to what we predicted. It wasn't even close. It was like 48% and 2%. It just didn't make sense. So he didn't understand what or how this was occurring. So he kind of posed this to his students. He said, hey, there's something's happening to these fruit flies. I don't know what's going on. See if you can figure it out. So one of his students named Alfred Sturdivant was in his class, and he actually blew off his homework that night and figured out what crossing over was. So they didn't know about crossing over, and this is what his data was. His data was 48% and 2. It wasn't the nice 25, 25, and 25%. And what was happening is he was figuring out crossing over, but he was also figuring out something called linked and unlinked genes. So what it should be is this. It should be 25, 25, 25. You can see all the way around, but it wasn't. It was this 48 and 2, and he didn't know why. And it was because certain genes are going to be linked together, and this is because they're found closer to each other on a pair of chromosomes. So these alleles here, these genes, are going to be linked because they're going to be found close together on the same chromosome. That means it's going to have a harder chance, or there is just less of a chance that crossing over is going to occur between them. So in this case, the A's and the B's are going to be inherited more often together than not because they're so close together on the same chromosome. And it's because, again, they are close on a chromosome. Let's take a look. Which one of these is more likely to occur? The crossing over, this side or this side? Which one's more likely to occur? This one's more likely to occur because there's just more area for crossing over to occur compared with this side. There's just not that much area for that crossing over event to occur. So those genes are going to be linked. I'm going to go back here. So A and B are linked. Now, A and C are technically somewhat still linked, but they're still good. It's just a higher chance that the crossing over is going to occur between those two chromosomes. So linked genes, these are genes that are located on the same chromosome, and they are going to be inherited more often together than not. So you can see these genes here are linked because they're on the same chromosomes and they're very close to each other. I like to use the example of the red hair and freckles are linked because they are found on the 15th chromosome or 16th chromosome here, and they're inherited more often than not because they're found very close together. So people with red hair usually have freckles because they're inherited together. Now on the opposite end, unlinked genes are on opposite chromosomes, are not on the same chromosome. You can see here gene 1 and these genes are unlinked because they're on opposite chromosomes. They're not on the same chromosome, so that means they're going to independently assort and they won't deal with crossing over. Now, we call this change in DNA recombinant DNA, and this is where the DNA is going to flip-flop. This is because of crossing over. So you can see our non-recombinant chromosomes and our recombinant chromosomes here because of crossing over. Now, I'm going to kind of not really go into too much detail here. I'm going to send you to a Bozeman science video. The Bozeman science videos are really good with this part. It talks about genetic recombination, but basically in these videos, Bozeman science is going to show you how to work through one of these percentage or these recombinant charts and show where these genes are located on a chromosome. So jump to the Bozeman science video about genetic recombination and it will help you with this. Now we're going to step into the new kind of like part of this chapter. The first part really looks at Mendelian genetics and how things follow kind of simple rules. At the end there, you kind of saw that things started not to follow Mendel's simple rules and what you learned in like your 9th and 10th grade biology class. And those concepts and those ideas do still apply to all of genetics. However, not everything follows those simple rules. Especially at the end there, you saw where the dihybrid Punnett squares wouldn't really be effective because the gene linkages might show something different in the offspring, so the genes are linked. We're not always going to see just simple Mendelian genetics. Our next concept or our next kind of big area in genetics is called non-Mendelian genetics. This is where it's not as easy and it gets really complicated from here on out. So just understand this is where that things kind of don't follow everything that Mendel saw. Mendel wasn't wrong, but he just didn't get everything. Multiple allele traits. This is a very interesting portion of genetics. I'd like to start off with this because this applies to most of human genetics or most of human genes, where there's not just going to be a simple big B, little b. There's only two alleles. There's going to be multiple alleles. These are just traits that have just multiple alleles. So it's not just big B, little b. There are just many different kinds of alleles. This image shows this is the most basic image for this concept, but you can see there are many alleles, more than two, which means there's going to be more than two phenotypes, or just say genotypes, but this also can produce more than two phenotypes. So this is where it gets complicated. There's not just two alleles, two phenotypes, and that's it. There are multiple of each, which makes things just, again, more complicated. Your HLA system, which is your like how your body identifies your cells versus non cells, or your cells that aren't yours. This is an example where you can have millions of different genotypes, and this is why you can't just transplant one organ to another person, because there are many different types of these HLA genes. So you have to be compatible with the person that you're going to get that transplant from. This is something we didn't know about initially, but now we have much better information and understanding of. So this is just an example of this multiple allele system. Codominance. This is an example of when there are, again, multiple alleles. Not always, I should say, but in some cases there are multiple. But in this case, several alleles will be dominant to each other. In roan cows, this is an example of a roan cow. So you have your red cows, which are red RR, white cows, which are WW. When they have an offspring, it's called a roan cow. So it has an R and a W. So it's going to be a mix of both. So in this case, again, the roan cow is codominant, or the genes in there, the alleles are codominant to each other. So it's not like we only see one. This is a codominant example. Now, this might look like that blending hypothesis. However, it's not. It's not a blend of either allele. You see both alleles in this example. I'm not going to show you this, but this is just showing you, hey, does the codominant support the blending hypothesis? It doesn't, because, again, two roan cows will have 25% red, 25% white, and then 50% roan. So it's still not just going to be roan. You're going to get those red and white cows if you cross these. All right, human blood typing is an example of both multiple alleles and codominance. Since there are multiple alleles, you can see they have the A, the B, and the O. In this case, in AP, we use these letters, the capital I, capital A, capital I, capital B, and I. In regular biology, I just use capital A, capital B, and O because it's a little bit easier, but I want to show you here that it is an example of a codominance. I really honestly don't care if you use A, B, and O, as long as you understand both of these alleles are dominant to O in this case. But this is an example of both multiple alleles and codominance. I'm just going to skip this for now. You see here. Okay, so that means there's going to be different genotypes. For A blood, you have AA and AO. Since A is dominant to O, this person still produces A blood. B blood, big B, or big BO. Yes, this still produces B blood. AB blood, you can see AB. O blood, you have to have OO. So the only time you get O blood, you're going to have two recessive O alleles. So again, there is 4 different phenotypes here. However, there are multiple different genotypes. Now, I'm not going to go through these in class, or I will go through those with you in class, not here. Talking about the type O blood is the universal donor. This is because there are no antigens to react with. Type AB blood is the universal receiver. There's no antibodies to react with. Let's go back here. Let's actually look at this chart here and then go back. So O blood can donate to anybody. AB blood can receive from anybody. So let's go back to these antigens. All right, taking a look here. We have these antigens. We have these antibodies and antigens. The antigens are little, like I call them flags on the blood cells that let your body know what your cells are versus not your cells. A has obviously A antigens, B has B antigens, AB has both, while O actually doesn't have any antigens. People with A blood have B antibodies. These B antibodies will attack B blood. However, they don't have B blood, so they're okay. Same with people with B blood. They have A antibodies. Sorry, people with B blood have A antibodies, so that A antibody is going to attack A blood. However, people with B blood don't have it. People with AB blood don't have any antibodies, so they won't react with any. People with O blood have both A and B antibodies, so they will react with all of these groups. So this is why you can receive, keep on going through here. You can receive from anybody if you're AB, but you can donate to anybody with O blood because O blood doesn't have any antigens while AB blood doesn't have any antibodies. One more thing about blood genetics is we have this Rh factor, and we call it the Rh factor because we first identified it in the rhesus monkey. Again, this is more evidence that supports evolution. Now, it's not saying we evolved from monkeys, but we share common ancestry, and some of the genetics that we see, even in our blood, is the same. Now, we can't transfer blood from monkeys to humans, but we see some similarity between the genetics. If you are positive, that means you have, again, this Rh factor or these more antigens. If you're negative, you don't have them. Now, if you have the homozygous dominant, that means you have, so it's positive, positive. You have the Rh factor, you're positive. If you're heterozygous, you're also positive. So you don't know if you're heterozygous or homozygous unless you do a genetics test where you know your parents' genetics in some cases. But if you are positive, it means you have these antigens and you're either positive, positive or positive, negative. If you're negative, you have to have both of them. That means you're homozygous recessive. If you have a positive, it means you have to have it. So the positive in this case is the dominant trait, while the recessive, homozygous recessive here, it's the recessive trait. All right, here's a good image showing that positive and negative. So you can see the positives have that extra antigen, that rhesus factor, while the negatives don't. AB has both the A's, the B's antigens. A's only have A's, B's only have B's, and O's only have the Rh factor if they have it, or they don't have any if they don't. I love this this image. This shows you the difference between the different antigens. Our next type of non-Mendelian genetics is incomplete dominance. This is when the heterozygous genotype is going to have a phenotype that is between the two dominant and recessive traits. Now, this looks like the blending hypothesis, but I assure you it is not. In this case, I didn't mention they were dominant or recessive. It could be dominant or recessive. Most of the time it's not. Again, you have three different phenotypes here. You have red, you have white, and then you have the in-between that is the heterozygous. This looks like the blending hypothesis. However, if you cross two pink flowers in this case, you're still going to get red, you're still going to get white. The blending hypothesis states that these should all be pink. So this is not the blending hypothesis, although it does look like it. Another type of non-Mendelian genetics is called sex-linked genetics. This is where any gene that's going to be located on the sex chromosome is going to be inherited a little bit differently than we saw in the autosomal chromosomes. So up until this point, we've been looking at autosomal chromosomes 1 through 22, and it's been nice and simple with even amounts being with both sexes. However, when you get to sex-linked genetics, it's not that simple. In humans, most of these sex-linked genes are going to be found on that X chromosome just because it's larger. I think the X chromosome, I don't think I have it here. Maybe I do. The X-linked chromosome or the X chromosome is, I think it's like 1,500 genes, while the Y chromosome is a lot less. I think it's only like 100 genes. So just by chance, there are going to be more genes located on that X chromosome versus the Y. Now, in these cases, males only need to inherit one of these traits, whether it's dominant or recessive, to inherit that disorder. Females need to inherit, usually if it's the recessive one, two of these traits. So we see sex-linked disorders a lot more in males than in females. You can see here an unaffected father, a carrier mother. In this case, half of the boys will be have that sex-linked disorder because they only need one of these traits, where females need two. You're not going to get any case of these females having the disorder. Now, they'll be a carrier, but they won't be affected by this disorder. Now, I'm not going to go through the examples here. I'm going to talk about Y-linked disorders. Again, this is only affects males because it has to be passed from parent to offspring. And in this case, males can only receive this Y chromosome from their father. So much less often than the X-linked chromosomes, but again, only affects males. If a father has it, then all the offspring will also have it. Or I should say all the offspring that are males. We're going to stop right there. All right, continuing with 11.6, we're going to talk about polygenic inheritance. And like the name suggests, poly meaning many, this is where several genes are going to contribute to the effect of one phenotype. So it's not just one gene, one phenotype. This is where several genes will all have an effect on a single phenotype. Now, again, just like multiple alleles traits, this is where we're going to see most of our genetics fall into, most of human genetics, where it's not just one gene per trait. There are many genes that are going to influence one trait. For example, height. It's not like you have two different heights in humans. There are many different heights, and there are many different genes that will contribute to the height of a human. I think that's still going to be one of the examples we're going to use here. Now, what this shows us is that there are many different phenotypes. Then, so it's not like we have tall and short humans. We have many different sizes of humans. I think the next one, yeah, here we go. You can see that bell curve here. This is what we're going to bring that bell curve back. Yeah, there's that bell curve. Showing that there are many different phenotypes of humans. Now, I like showing this image because it's a little bit misleading. It makes you think that women maybe be taller than men. No, obviously men are a little bit taller than women. It's showing us that there is so our height is down here. There is a wider variety of men's heights. However, men are taller than women, and more women will fall under this category of the average. And this shows us that standard deviation, what we talked about in the beginning of the class, many months ago. Now, I'm going to skip ahead here because there are many examples of polygenic inheritance. I'm just going to skip just to the more important things here. Incomplete penetrance. This is an example of when a genotype won't produce the phenotype. So sometimes, yes, you can inherit something, but not show the phenotype. And there's a lot of kind of mystery here where it's like, well, why doesn't it? Well, there are many different things, many different factors, many things that we just don't know within genetics. So this is when you inherit something but you just don't have it. So you can see the BRCA1 gene. This is when somebody will inherit this. This is a breast cancer gene, and you could have this gene and not develop breast cancer. 80% of the people who develop it will, but not all of them. And there's a lot that goes on with this. So there are many factors to these genes, and I like to show this just because it's not as easy as Mendelian genetics kind of makes it seem. Tongue rolling is an example of this, where you could have the genes for it, but just not show that phenotype. Pleiotropy. This is when one gene will have, so it's kind of the opposite here, as where one gene will have an effect on many different phenotypes. And this is because that one gene is very important to the phenotype of the organism. So you can see here, one gene having many different phenotypes. An example is like people with albino skin. They have one gene that's affected, but they have many different phenotypes that is affected because of that one gene. Or dwarfism, having one gene be affecting many different phenotypes. Last part of 11.6, and yes, I know there's been a lot of information with non-Mendelian genetics, but there are just so many different examples of non-Mendelian genetics. Is a term called epistasis. This is where there's an interaction between two genes. I saw this image once and I thought it was great. It helps us understand that one gene may influence the effect of another gene. So like gene A in this case is controlling gene B, where this gene needs to be activated so this gene can also be activated. And let me show you an example here with dogs. So you have this melanin gene in dogs, that's the A allele, and the quantity of that pigment or that gene is produced here in that B allele. So what happens is, again, we get more phenotypes than just one or two different phenotypes. These types of dogs, labs. There we go. Remember these labs. If they have this gene turned on, this activated, and they have a capital letter in the second one, they're going to be black labs. If this gene is activated, however, they produce the brown gene, they're going to be brown labs. However, if this first gene isn't activated, it doesn't matter what this second gene is, they're going to be yellow labs. This is because A controls B. If A is turned off, then B will also be turned off. That means you're not going to get any pigment. So this is where you can get several different phenotypes because one gene essentially controls the next gene. All right, our next section here is going to be on pedigrees, and this is kind of like a review of everything that we've talked about within genetics, because we're going to be able to see Mendelian and non-Mendelian genetics in these pedigrees. And pedigrees is a pedigree is a kind of like a family tree. This is a pedigree, and it traces the inheritance pattern of a family. Now we can come back and talk about these P, F1, and F2 generations from section, I think 11.2 or 11.3. But basically, this shows us how genetics are passed from parent to offspring. Now, in these pedigrees, males are going to be square, females are going to be circles. Shaded circles and squares are going to be affected. That means they have whatever disorder or trait that you're looking at. Carriers, these don't express the trait or phenotype or whatever, but they still have one allele. So they're half shaded in. Another name for a carrier is heterozygous. So whenever you see a circle that's not shaded, that's a female. Circle that's half shaded, that's a carrier female. That's, again, heterozygous. An affected female is shaded in, and on the opposite end here, male's square or male it's square, unaffected, half shaded in, carrier male, heterozygous, an affected male, shaded in. Now, lines between individuals represent that couple has produced an offspring. Lines and the circles and squares below show that they are the offspring of the parents. And like in this case here, these two would be siblings. And the generation number here, like generation 1, generation 2, you could also have this as P and F1 if you'd like. I've seen them both, and there's a lot of different types of pedigrees, but I'm kind of showing you the basics of these pedigrees. Right. If big A, and big A are both possible, and we don't know which one it is, the answer big A question mark is the only correct answer. You can't definitively say in this case what this offspring is going to be. This is big A, little a. This is big A, little a. Now, it doesn't show it's half shaded in, but if we know that this individual is affected, that means these have to be carriers. Now, if these are carriers and we do the monohybrid Punnett square in our head real quick, this could be big A, or big A, little a. We just don't know. So this one's going to be a big A question mark. So sometimes this big A question mark is the only correct answer. Now, we can show in these pedigrees an autosomal dominant pedigree, an autosomal recessive, and each is going to be different in how they are inherited. And you're going to get practice with this with your packets, but we can also show this in sex-linked genetics, where you have the X-linked dominant and X-linked recessive. So yeah, we can bring those back and show those in a pedigree. We can also show a Y-linked inheritance pattern in a pedigree. So we can do a lot with these pedigrees, and it's based on the inheritance pattern of that pedigree. The last section of this chapter, and actually unit, is a very quick one. I just like to talk about the environmental factors on genetics. I like to talk about something called phenotypic plasticity. This is where the environment will have an effect on a phenotype. So it's not like just its only traits. The environment will affect how a phenotype is expressed. It's not as perfect. We see this a lot in human populations. Let me show you an example here where you have two different twins who have the same exact DNA, but obviously they express that DNA just a little bit differently, and it's due to their environmental factors. You can have seasonal fur color. So you could have organisms that have different colors of fur based on how warm or cold it is. You can see the phenotypes of these different insects developing differently because of the temperature. So you have a lot of different types of phenotypes, and it's based off of sometimes just the environmental factors that are applied to an organism's phenotype. So it's not only the genes, sometimes it could also be the environment that controls the phenotype.